Hi everyone. We're going to be talking about working with nucleic acids today. Working with nucleic acids is a common activity in research and development. There are various techniques and procedures that are routine and will be discussed in this upcoming lecture series. The first important set of techniques involves isolating and purifying DNA or RNA for further manipulation in the laboratory. These methods involve lysing cells to release the nucleic acid and then separating the nucleic acid of interest from the other cellular components. The topics to be covered in this section are phenol chloroform extraction, salting out, silica exchange resins, antibody-coated magnetic beads, the use of Helix beads, and concentration of DNA using alcohol precipitation of DNA, and spin columns such as the Microcon 100. What are the essential components of a DNA extraction procedure? When attempting to isolate purified DNA for downstream analysis or manipulation in the laboratory, there are several things that must be considered. First, the method should be performed to maximize the recovery of DNA. Second, inhibitors of the enzymes needed for downstream work must be removed from the DNA solution. Third, nucleases that could destroy your DNA must be removed or inhibited from acting. Finally, the DNA must not be sheared or nicked. The quality of the template must be preserved as best as possible to guarantee functionality in the downstream uses. The average eukaryotic cell contains enough DNA to stretch out about 6.5 feet and has a mass of about 6 picograms of DNA. The gametes, such as sperm, would contain half of that, so about 3 picograms. Considering blood as a source, there are between 5 and 10 million white blood cells per milliliter of blood. And since there is a million picograms per microgram, the recovery of DNA from a milliliter of blood is theoretically around 60 micrograms of DNA. Plasmid preps vary in efficiency or recovery. The copy number per cell of plasmids also vary, so that the final purified plasma amount will also vary, but a typical yield would be near 5 micrograms per milliliter of starting culture. More or less may be expected once you are familiar with your plasmid and your method. Visualizing DNA in agarose gel requires the use of dyes to stain the DNA. There are various dyes available. One of the most common is ethidium bromide, which is also a mutagen and must be handled appropriately. To visualize DNA, the dye, the size of the fragment, and the gel type will influence the ability to see the bands, but a minimum of 10 to 20 nanograms per fragment in a 2 millimeter wide lane would be needed. Larger lanes and smaller fragments will likely need more to see clearly. PCR reactions can be used to detect the presence of a sequence or may be needed to amplify a sequence for downstream uses. The sources of the DNA, for example genomic versus purified plasmid, will influence the starting concentration requirements. The conditions would need to be optimized for the different reactions. While it is theoretically true that only one intact template sequence would be needed for a successful amplification reaction, 1 picogram to 1 microgram of DNA template will be the typical starting requirement in the laboratory. Transformation reactions typically use between 100 to 100 picograms to 100 microgram. Transformation reactions typically use between 100 picograms to 1 microgram of plasmid DNA for a transformation efficiency of up to 1.5 times 10 to the 9th transformants per microgram. DNA extraction procedures may involve the use of organic solvents or silica exchange resins. The silica exchange resins are readily available in kit formats and do not require the use of solvents in a fume hood, so they are the preferred method by a large number of researchers, especially for smaller DNA molecules such as smaller plasmids. Non-organic salting out of DNA may also be used and will be discussed shortly. A classic method of purifying DNA from other cellular materials, especially proteins, is phenol chloroform extraction. While this method is good for purifying large DNA molecules, this method has largely been replaced due to the toxic nature of solvents involved when smaller DNA molecules require purification. Working with phenol chloroform requires the use of a fume hood, personal protective equipment, and proper disposal of the solvents. When conditions are properly monitored, the method involves the separation of an aqueous layer, which will contain your DNA, and a solvent layer, which will contain the cellular protein. DNA can be further purified from other cellular components, including salts, by isopropanol or ethanol precipitation, to be discussed shortly. In the organic extraction using phenol chloroform methodology, a non-ionic detergent is used to suspend a cellular pellet and will lyse the outer membrane but leave the nuclear membrane intact. Salt, buffer, and EDTA are also included in this first solution. Importantly, the EDTA is a chelating agent of divalent cations. A chelating agent binds to these ions and makes them unavailable. The ions are needed by the enzymes nucleases, which would degrade your nucleic acid. Thus, the addition of EDTA is a protective agent for your DNA. After pelleting the nuclei of the cells, a solution containing SDS will be used to lyse the nuclear membrane. SDS and sometimes the addition of heat will denature proteins, which are contained in your solution with the nucleic acids. Incubation with proteinase K is often included because it will digest a wide variety of protein. It is very active, tenfold more so, on denatured protein, so the proteins denatured by SDS and heat are highly susceptible to the breakdown with this enzyme, furthering your purification. Phenol and chloroform are often used together in a protocol to purify nucleic acid. The standard method involves one extraction with phenol, one with a 1-to-1 mixture of phenol and chloroform, and a final extraction with chloroform. Abbreviated methods can be used omitting one or more of the extraction steps, but the resulting nucleic acid will not be as pure. This standard procedure takes advantage of the fact that extraction with two different organic solvents is more efficient at removing various proteins than a single solvent. It also allows for the removal of all traces of the corrosive phenol. However, as previously explained, the entire method involves solvents that must be worked with under a fume hood and be disposed of properly. Of note are the facts that phenol is usually equilibrated with a buffer that contains 0.1% hydroxyquinoline and 0.2% beta-mercaptoethanol. These are added as antioxidants. Another advantage to this is that phenol will take on a yellow color, making it easier to distinguish the solvent phase from the aqueous phase during the extraction. Chloroform typically is mixed with isoamyl alcohol with a ratio of 24:1 to prevent foaming. So when making the phenol-chloroform mixture, the ratio is 25 parts phenol with the antioxidants to 24 parts chloroform to 1 part isoamyl alcohol. The advantages of phenol-chloroform extraction are that high molecular weight double-stranded DNA can be purified to relative purity. The disadvantages of this method are that compared to other methods, it's time-consuming. It requires multiple transfers, which increases the risk of contamination of your nucleic acid, and it involves smelly and hazardous solvents. There is a method called salting out that removes digested proteins with high concentrations of lithium chloride instead of organic solvents. However, if the salts are not adequately removed, the DNA will not be useful in downstream analysis or manipulations, as the salt will inhibit later results or alter your results. For example, bands will not run true in gel electrophoresis, altering interpretations of RFLP tests and of the DNA samples. Did I say that right? For example, bands will not run true in gel electrophoresis, altering interpretations of RFLP tests of DNA samples. There we go. In the salting out extraction procedure, the initial lysis followed by proteinase K digestion is similar to the phenylchloroform procedure. However, lithium chloride is added after treatment with the proteinase K at a final concentration of 2.5 molar. This is incubated on ice. Proteins precipitate and they can be separated by centrifugation. The precipitated protein pellet is separated from the solution which contains the DNA. The lithium chloride is separated from the nucleic acid with an alcohol precipitation step. Washed and dried DNA can be resuspended in an appropriate buffer such as TE. In the presence of salts and a specific pH, nucleic acids can bind to silica. This feature has been exploited by manufacturers to produce commercially available kits which contain silica within columns. The kits will also include lysis, wash, and elution buffers such that DNA bound to the silica can be purified and then eluted into a collection tube. These columns and kits can be purchased in various sizes such that one can perform small-scale mini preps when only a small amount of DNA, such as less than 20 micrograms, is needed, or large-scale maxi preps when much larger amounts of DNA, like 500 to 800 micrograms, might be needed for manipulation. The advantages of a silica-based extraction method is that it is relatively quick and you could obtain highly purified DNA. The disadvantages are that the commercially available columns are typically more expensive than the solvent and the salting out procedures, and the procedure requires multiple transfers between tubes, which increases the risk of contamination. Another useful method for obtaining purified nucleic acids involves commercially available magnetic beads. These beads are coated with antibodies that specifically bind to DNA. The beads can be separated relatively easily from the other cellular components with the use of magnets. The DNA is then eluted from the bound antibodies in purified form. The magnetic bead procedure has been developed into an automated process used by some high-throughput laboratories. Samples are loaded into wells on plates containing the beads. The wells can be treated with various solutions to lyse cells and wash the DNA. These solutions can be delivered via a robot. This method can provide large numbers of PCR-ready samples in a relatively short period of time. The purified DNA will be free of inhibitory substances such that the PCR reactions are reliable. The advantages of the magnetic beads is that the procedure is very quick and it can be automated to produce highly purified DNA. This can be very useful for dealing with samples of liquid blood. The disadvantage to this method are that the beads are very expensive compared to the other methods of DNA preparation. Also, while excellent for liquid blood, it's not as useful for dried blood or stains that might be analyzed in a forensic type laboratory. These samples would need to be pre-treated to release the cells into a solution that could then be assayed. Helix resin, made by the manufacturer Biorad, is another resin often used for preparing DNA samples for PCR reactions. This resin is a chelator of the metal ions that are required of the enzymes that degrade DNA, the nucleases. Helix 100 resin is added as a 5% weight to volume working solution. The copolymers of the resin act as chelating groups of polyvalent metal ions such as The copolymers of the resin act as chelating groups of the polyvalent metal ions such as magnesium ions. Magnesium ions are required cofactors for nucleases that degrade DNA, so by removing these ions, nucleases cannot function and the DNA is protected. An example of a preparation using chelates would be as follows. The 5% chelates is added to a small amount of blood and it's incubated at elevated temperatures such as 56 degrees Celsius to lyse the cells. The sample is then boiled for 8 to 10 minutes, which will denature protein and cause the DNA to dissociate into single strands. This is fine for PCR reactions, which require DNA to be denatured. The chelates beads with bound ions are centrifuged to pellet them, and the supernatant containing the DNA can be used as the template for PCR reactions. Caution must be taken not to accidentally transfer residual chelates with the template DNA, since magnesium is required by Taq polymerase, and including chelates with the template will thus inhibit the PCR reaction. The advantages of using chelates to acquire DNA for a PCR reaction is that it's relatively easy to use and it's relatively fast. A small snippet of cloth with a stain can be directly used to acquire the DNA sample. Since only a single tube is used to prepare the sample for analysis, it minimizes the chance of contamination when you transfer tubes. This method focuses on removing PCR inhibitors, and thus it's advantageous for this method. But the disadvantage is that since this method results in single-stranded DNA, the method's not useful for other types of DNA analysis. DNA can also be concentrated using membrane filters that allow for retention of molecules larger than a specific size. Smaller molecules pass through the filter, and the larger molecules are retained in the upper chamber. The Microcon series are available from the manufacturer Millipore. The Microcon filter units are an alternative to alcohol precipitation of nucleic acids. These units can be used to purify proteins of a specific molecular weight, as well as nucleic acids. They come in different varieties depending on the size, e.g., are you purifying genomic or plasmid DNA? So depending on the size of molecule that needs to be concentrated, you choose a specific filter type. They can achieve a high recovery of your sample and are used to concentrate solutions of DNA that contain low concentrations that would be difficult to recover using traditional alcohol precipitation. This method is great for removing primers, label, or other leftover components of a previous reaction prior to using the DNA in a subsequent reaction. Finally, we'll talk about purifying DNA by alcohol precipitation. Alcohols, such as isopropanol and ethanol, are often used to precipitate DNA in order to concentrate it from a more dilute solution, and also to remove contaminating salts. This technique requires the presence of a moderate amount of salt and works well with nanogram or higher amounts of DNA concentrations. The precipitated DNA is pelleted by centrifugation, and the supernatant is removed from the DNA pellet. Ethanol is preferred, but it requires a higher volume of DNA precipitation, so sometimes isopropanol is used, followed by washing the pellet with ethanol. Ethanol evaporates more readily than isopropanol, so a dry DNA pellet is more readily resuspended in the desired buffer following an ethanol precipitation or wash. The critical variables for obtaining precipitated DNA with alcohol precipitation is that the purity of DNA in solution, meaning how little contaminating protein has been carried through your procedure. 2, the molecular weight. The larger fragments are easier to precipitate. Less than 200 base pairs in size is difficult to precipitate with alcohol. 3, the concentration of your DNA solution. 20 nanograms per milliliter or higher is needed for a good recovery rate of your DNA. 4, the volume of alcohol. At least 2 to 2.5 times the solution volume is required for an ethanol precipitation, and at least 0.7 to 1 volumes is required for an isopropanol precipitation. These alcohols can be added to precipitate the DNA, and you need to keep in mind your total volume to be held in the vessel that the DNA is then to be centrifuged in. 5. The speed at which the DNA is pelleted. You'll need at least 14,000 times G to pellet your DNA. So as a means of comparison, isopropanol is used to precipitate DNA when a smaller volume is needed to fit in a vessel that is used to centrifuge your DNA, to pellet your DNA. However, it does not evaporate as readily as ethanol, such that a 70% ethanol wash of the pellet is usually required before the DNA can be dried and resuspended in a storage buffer. Ethanol is thus preferred for precipitation, but you'll need a larger volume to precipitate the DNA, so care must be taken so that your total volume required to precipitate will fit in your vessel. And finally, you need to be able to resuspend and store your DNA. DNA is usually stored in a buffer such as tris EDTA, called TE buffer. The presence of EDTA chelates magnesium ions that are needed for the nucleases that degrade DNA and is thus protective. Each molecule of EDTA will chelate one molecule of divalent magnesium. EDTA may be included as a 1 millimolar concentration or a 0.1 millimolar concentration, depending on downstream use. Transfer of larger volumes of EDTA in the higher concentration may inhibit downstream reactions, such as PCR, that require magnesium, or ligases, which also require magnesium. The slightly basic solution prevents depurination of DNA, which is an acid-catalyzed reaction. Storage in refrigerated temperatures is fine for short periods, but DNA is frozen for long-term storage. Laboratory freezers should not have a freeze-thaw or a de-icing cycle, as this will cause DNA to degrade over time. Lyophilization, which is a freeze drying, is best if very long-term storage of the DNA is needed. Lyophilized DNA can be stored at room temperature in a protected container for years, as it is stable. This concludes the first series of working with nucleic acids. Thank you.